Okay, now let's talk about antibodies. Before we can talk about what an antibody is, we need to know what an antigen is because antibodies work against antigens. So let's first look at what the antigen does. And let me just write this out here. So an antigen is any substance that has a potential. So it's any substance that has a potential to initiate or to start the immune response. Okay, so this could be any foreign antigen getting into your system or in your system. It could be a bacteria, that could be a virus, it could be pollen. Okay, you could cut yourself and that starts an infection. Okay, that starts the immune response. So an antigen is any substance that has the potential of starting or initiating your immune response. So now let's look at what an antibody is. Now an antibody is, remember when we were talking about those B cells, those plasma cells? So it is produced, or it's a substance produced by plasma cells that immobilize antigens. Okay. Now, an antibody doesn't directly destroy the antigen. It essentially tags them for destruction. Okay, so it just marks them or tags them to attract other immune cells to then kill it. Okay, so that's what that is doing. Let's look right now. Let's look at the antibody. Okay, so this is the basic structure of an antibody. You've seen me draw this on B cells briefly, that Y-shaped thing, okay? But this is essentially what the antibody is. So let's go through the various parts of this, and then we'll talk about what the antibody is actually doing. So let's look at a couple different things first. That's your basic Y shape. This region is called the arm. This is the, let's do this, just do this. That's the stem, okay, on this one. Now, a couple different things. This region here, that's a variable region. And we'll describe all those here shortly. This is the constant region. These are, now let me just point them out to you in case that you'll need this information at some later time. You can see this region here, that's a light chain. And these are heavy chain. Now the stem, this region for the stem, excuse me, is also known as the FC region. And we'll talk about that shortly, okay? What else are we missing here? We have your antigen binding site. Okay, that's going to be a big one. Antigen binding site. And there's actually two of those, okay? That's that binding site and that's binding site. Now, the antigen binding site is very specific to an antigen, okay? So they're just not generalized. So this is going to be specific to one specific thing. Go back to those previous videos on figuring this out. Here it's just kind of shaped as a circular shape for visual representation. Sometimes for visually on receptors that I'll do a triangle shape or square shape or something like this. But these are all very unique shapes or very unique structures for a specific antigen. Okay, so what we're looking at now, now the antibody can work against or is effective against antigens by one of two different ways. Okay, it will work with the variable region. So the variable region, which includes the antigen binding site, the variable region can bind with, it's a very weak binding, so you're like a hydrogen bond or an ionic bond, something along that line. It's very weak things, and that either the antigen binding site or the FC region of the stem, okay, so stem or FC region, that can also immobilize antigen. So your variable region or the constant region can bind with and immobilize the antigen, which then prevents them from doing or initiating the immune response. And now there's 6 different ways of that happening. 3 ways are associated within the variable region in the arms, and then 3 different ways for the constant region on here. So let's look at what we're doing. Let me just look. I just want to make sure I get this good. Okay, so the different things that let's do the variable region first on how that can limit the effect of the antigen. Okay, so we have 6 different ways. 3 ways for the variable regions, an additional 3 things for the constant region. So how this antibody can prevent this is we can neutralize the antigen. Okay. And what this is doing is the antigen will have the active site that could infect the other cells, and what the antibody does is just kind of blocks it. Okay, so if the active site is being blocked, it can't do its job. Okay, another thing we can do is agglutinate. How do you spell agglutinate? L-U-T-I-N-A-T-E. Okay, it will agglutinate the antigens. On this one, go back last semester when we were talking about blood typing. And we have different antigens and different antibodies. And if you transfuse the wrong type of blood, we call that blood will become agglutinated, or it will agglutinate. That's what this is talking about. Okay, so it's clumping them together, and it prevents them from doing what they need to do. Another one would be precipitating, or it precipitates out. And what it does is it binds or cross-links multiple antigens. So you're forming antigen-antibody complexes, and it precipitates out of solution. So it kind of falls out of liquid, if you think of it in those terms. And when it does that, other immune cells can come in and then destroy that. Now, those are typically of your variable region. And for the constant region, we have 3 other ones that can do. It can be part of your complement system. Okay, now this leads into MAC. Remember that one? Okay. Now complement, if you recall from the previous video, this one has a series of cascading steps leading up to MAC. Okay, and within that series of steps, we have other things. We can have other inflammatory reactions, opsonization, all these different things on it. So since we said opsonization, one other one would be that, how do you spell opsonization? Opsinoation, opsonization. And opsonization essentially allows or stimulates phagocytosis of that antigen. So it binds with the antigen, then it is attracting other cells that have phagocytic properties to then engulf it and then destroy it. So this is going to be dealing with the phagocytosis. So it increases phagocytosis. This is okay. And the last thing will stimulate natural killer cells. And this one is going to lead to apoptosis of cells. Okay, so those are your different things on this. So with that, now let's look at immunoglobulins. Where's immunoglobulins? So let's do this. Now, what are immunoglobulins? Immunoglobulins are antibodies, and there's 5 classes of antibodies that we're going to look at, and they're abbreviated immunoglobulins, abbreviated IG, and we're going to have another letter after that. So there's going to be 5 classes of immunoglobulins, and you can remember these with the acronym of GMADE. Okay, so GMADE. Okay, so we're going to have IgG. That's going to be your first class, and this is your primary one. This is where most of your immunoglobulins are going to be IgG. So this is going to be, 80%, 75 to 85%, 75 to 85%. And this is found typically in your lymph fluid, your serous fluid, peritoneal cavity. This is a very small antibody, and it can cross placental barrier. Okay, so remember again, last semester we spoke of hemolytic disease of the newborn. This is where, the Rh factor issue, if you're not familiar with that, go back and review that information. But these antibodies are small enough to cross the placental barrier and then go after that blood type in the fetus on this one. So this is the hemolytic disease of the newborn, also known as erythroblastosis fetalis. That's it, erythroblastosis fetalis. The other class, or another class, is IgM. So IgM, and IgM is going to be the reaction that you get with blood typing mismatch. Okay, so this is what happens. So this is going to cause agglutination, because remember above we had agglutination up here. So IgM would deal with agglutination, and this is blood mismatch. So your blood would agglutinate on this one. We're going to have IgA. And IgA you're going to find in mucous membranes, breast milk. You're going to find them in your tonsils. You will see a heavier concentration of these in your respiratory and GI tract. So, although I have not heard yet, I would speculate that with this COVID virus, COVID-19 going around right now, which is primarily a respiratory issue because people end up with pneumonia and those sort of things, I suspect that those individuals would have an elevated IgA count in the acute phase of the infection. Why don't you look it up, see if you can find any research on that to see if COVID-19 is associated with an elevated IgA count. That'd be interesting to know. So I'll let you guys deal with this one. So IgA, so this is what? Mucous membranes. So what I say, this is going to be tonsils, mucous membranes, tonsils, breast milk. So respiratory and GI tract on this one. Another one, let me raise this up a little bit. IgD. These are going to be the receptors on the B lymphocytes. So these are found on those B cells. Because remember, I would always draw that Y structure on a B cell. That's IgD. Okay. And the last one is IgE. And this one's going to be dealing with allergic reactions. So these are going to be allergic reactions. So you're going to imagine that's going to release histamine. And this is going to be allergic reactions, also parasitic infections. And histamine, so what cell type releases histamine? That's associated with parasitic and allergic reactions. That's going to be eosinophils. Okay, so that's a quick down and dirty view of antibodies, what they are, what an antigen is, what are immunoglobulins, what their basic functionality is on this one. Next up, we are going to look at immune memory on this one, and we'll just focus this in, and I think that might be it for immunity. Not sure, but I think that might be it. Thanks.